other reason that you have these phylogenies is because species share evolutionary relationships, right? And so that means each of your single species estimates are not statistically independent because species share an evolutionary history. And we can take advantage, and so we can estimate this relationship between phylogeny and traits by phylogenetic signal here. And so what you can see, so we've estimated phylogenetic signal and heat resistance in the Drosophila species, and we have a significant signal. So once again, the lambda estimate of signal, 1 being like complete phylogenetic signal and 0 being no relationship between phylogeny and traits. But we can also use this phylogenetic signal as a way to kind of study how traits have evolved across the phylogeny and make predictions about which groups of species may or may not be at greater risk to climate change. And so what we found was, I think, kind of surprising. It was, oh, okay, now my that's all right.
Try that now.
And so what we found with the Drosophila phylogeny was that only a handful of species have actually evolved high heat resistance. And once again, these are our desert species. And so this kind of suggests to us that these desert species have been able to kind of move into these environments because they have the capacity to evolve higher heat tolerance, right? But the remaining rest of the Drosophila phylogeny, they don't have that capacity. And so this suggests that there could be these limits in the capacity more generally of Drosophila species to evolve high heat tolerance. But some of you also might be thinking, well, there's kind of two aspects of evolution, right? So there's genetic evolution, but there is also phenotypic plasticity, right? So each of these data points, different species will have different capacities to respond to the environment via phenotypic plasticity. And so here I'm defining phenotypic plasticity as a single genotype responding differently in two environments. And because we don't work on clones, we often kind of look at the population level. So it's not single genotypes per se. And so here is just an example of heat resistance where we don't have any phenotypic plasticity. And this is what phenotypic plasticity would look like in heat resistance. And I think the phenotypic plasticity people are very descriptive in their language. So I'm defining here adaptive phenotypic plasticity. And what I mean there is that the change in phenotype is increasing fitness in the given environment. And so we don't actually, and this is very rarely done, do we go out and actually estimate the change in fitness in that environment. But we are assuming here that if we rear flies under warmer conditions and they have higher heat resistance, that this is indeed an adaptive response. And because, yeah, it's we're very nuanced in this plasticity world, I also wanted to just get some terms right with you as well. And so people often think of plasticity in depending on when it's measured. So you have developmental plasticity or developmental acclimation, and so that's rearing egg to adult at different environments. Then we have adult acclimation, which is exposing adults during, usually for days or weeks, at kind of a more benign temperature. And then we also have rapid hardening, and so this tends to be a more extreme kind of exposure that you give an organism for minutes or hours. So I think I've lost my capacity to change my slides again. And one of the reasons that we're really interested in phenotypic plasticity is because people have proposed that phenotypic plasticity could be really important for species responses to climate change. And so when we think about evolution, or genetic evolution at least, or genetic change, we often think that that's going to be quite slow. And if we think about Drosophila melanogaster, it's highly likely that they're going to need some kind of de novo mutations, to be able to increase their heat resistance significantly. And so people think that, plasticity could buffer species while climate change can genetic change can catch up, so to speak. So we were really interested in this question of just, well, how much can developmental plasticity, so developing flies from egg to adult under different temperatures, how much can it actually shift heat resistance? And so this is some work that we did in 2 populations of Drosophila melanogaster, and we had a range of different developmental temperatures, which you can see on the x-axis, and heat resistance on the y. And what we found is that over a 14-degree temperature range, we could only increase heat resistance by about 1 degree. And if you kind of look at that figure in a bit more detail, what you might notice is a lot of that shift in heat resistance is actually at the cooler end. So when we start looking from about 24, 26 degrees onwards, we're actually not getting that much of a shift in heat resistance because of these developmental temperatures. And so we were interested in whether this was just a pattern that was common in Drosophila melanogaster, or perhaps something, and whether different Drosophila species might be a little bit different. And so we went out and we estimated developmental plasticity in 20 or so Drosophila species. And so this graph, I'll just explain it a little bit because you need to read it slightly differently. So on the y-axis, we have heat plasticity. So this is really just the difference of your heat resistance at 27 degrees minus 20 degrees. And so it's your plastic capacity. And then for reasons that I won't get into, we have environmental temperature that the species are found with on the x-axis, and that blue line indicates 0 plasticity. So developmental acclimation hasn't actually shifted your heat resistance at all. And actually what we found was in 4 species, warmer developmental temperatures actually cause a decline in heat resistance. So we're kind of getting this maladaptation plasticity occurring. And we didn't really see that much plasticity really happening. And across this 7.5 degree developmental temperature range, we're only getting a less than 0.3 degree change in heat resistance. And I also want to highlight that those 4 species tend to be tropical species as well. So they're really not doing well when we develop them under those warmer temperatures. Okay, so I probably sped through that way too quickly, but I just want to kind of sum up what we've done in Drosophila space. So we've found that environment is an important predictor of heat resistance. So it suggests that selection is acting on heat resistance, and heat resistance is really important for shaping where species live. We've shown through kind of breeding design experiments and selection experiments that there doesn't seem that much capacity for heat resistance to respond to selection, at least in Drosophila melanogaster. And then when we go over to the big species comparisons, we can see that we have this strong phylogenetic signal, and only a handful of species have really only been able to evolve high heat resistance. And I think, yeah, what we need to do now is really to pull out some extra species, some really heat-tolerant species, cold-tolerant species, and start selecting on them and seeing if we can increase their heat resistance. And we also show that at least developmental acclimation is probably not going to contribute that much to buffering species against climate change. And so one of the things that I often get from reviewers is, well, this is just Drosophila. Why do we really care? And I mean, they have a point to a certain degree. Like Drosophila is a really great model organism, and we can do lots of really cool stuff in the lab. But insects have really diverse ecologies, and whether this species is a really good model for understanding climate change responses is a valid question. And so this kind of brought me to start working on this slightly more charismatic fauna, the native bees in Australia. And so the question that we had was just that simple one, starting back from the start: how does climate shape native bee distributions? And because I still feel very new to the bee world, I thought I would also, I'd take advantage of being able to show some beautiful pictures of bees, but I thought I'd give you all a bit of an intro, because when I first started working with native bees, I think it was these kind of bee facts that I was just totally fascinated about. And so, sorry, in Australia, we have maybe 1,600 to 2,000, but no one knows. We have a lot of native bee species, and this is part of our problem. We don't have that many people working on native bees in Australia. So we've only got about 4 taxonomists, and 3 of them are hobbyists, right? So that's all we have. And so when you ask someone in Australia what a native bee is, they will think about the beautiful European honeybee, and they're actually quite surprised to learn that is indeed an invasive species. In Australia, we don't have bumblebees. We do have, our only social bee that we have are our stingless bees, and the rest of the remaining, and so we only have 12 stingless bees, so the rest of the 1,600 or so bees are solitary, communal, and so I was really fascinated to learn that, sociality isn't you're either social or you're solitary, that sociality is really a spectrum. And so we get these amazing diversity of spectrums of sociality in bees. And so I work on a species that is socially plastic. So it might be solitary, or it might nest with its sisters. And then you have these hierarchies forming in these communities. And we also have a diversity of nesting strategies in native bees. We have stem nesters, and they make their nests in these kind of pithy stems. And then we have our cavity nesters, and so that also includes our stingless bees. And so they either nest in kind of man-made cavities or big kind of wooden logs. And then we have our ground nesters. And I'm not actually sure about the native bee facts in Europe, and maybe these are all kind of global, but in Australia, at least 70% of our native bees are our ground nesters. And so I often see people put out these bee hotels, and I'm like, and of course, we all know they're really important pollinators. They pollinate our wild native plants, and they're really important agricultural pollinators as well. And so in Australia, we don't have bumblebees, and so some of you might know this, but bumblebees are buzz pollinators, and they're really important for pollinating tomato crops. And so honeybees can't buzz pollinate, so our buzz pollination really comes down to our beautiful native bees. And there's research showing that native bees are playing an equally, if not more important role in pollinating agricultural crops. And so first of all, I wanted to introduce you to the people that have worked on this project, because it wouldn't be possible without them. And in particular, I wanted to highlight up on the left-hand side, and this is Dr. Loeschcke, and she has been the person that has taught me everything about bees, and she's an amazing bee researcher, and she actually named our group Team Let's Cook Some Bees. And so to do this bee research, we needed to go out and collect bees. So unlike Drosophila, we can't rear them in the lab, and so we needed to go on a really big field trip and collect bees. And so when I say we, it was really my postdoc, Carmen De Silva, and her partner did this really amazing field trip where they went along the east coast of Australia. And we're really fortunate in Australia that we have this kind of beautiful temperature gradient. And so they estimated heat resistance, cold resistance, and desiccation resistance in the field at all these different sites. And we also, I'm really interested in desiccation resistance as well. And so we also had some desert environments in there as well. Bees are hard. They are really hard. They're hugely cryptic. And I think this has been one of the biggest challenges of this project. And so we were lucky to have kind of one of the hobbyist taxonomists agree to come on board onto our project. But I don't think he really appreciated when he said yes, that we would be handing him 10,000 bees to ID. So it's been very challenging. And, and we have some groups of bees that don't even have morphological keys. So it's just really, really challenging. So I thought I'd just give you a quick example. And because they're hugely cryptic, right? When we did this project, we did what I call the smash and bash approach. Like we just went out into the field and we hoped that we got something. And so this is what we would do. We would find a eucalyptus tree that was flowering, and we'd use these really long nets. And then we would get all these bee species. We would bring them back to our makeshift labs and Airbnbs. We never told our owners. And then we'd estimate all our different traits. And then we'd bring them back into the lab, and our amazing taxonomist would do a lot of the species IDs, but a lot of them, the ones that we could only get to morpho species, we've done some DNA barcoding as well. And we've tried our best to get as much distributional information for each of the species that we've got that we can, but it is not as good as the stuff that we have for Drosophila. And then we've extracted environmental data from the distribution. And because not many people have really worked on bees, we had to create a phylogeny as well. And so we created an ultra-conserved phylogeny with a backbone of 80 species. And then we're filling in that phylogeny with CO1 barcodes just for the remaining 20, 50 species. And so in Australia, we have 5 main families of bees. We have the Apidae, Macroecological, Collididae, Polyptidae, and we also have Stenotrididae, but that is a really rare family in Australia. And so of the 10,000 bees that we collected, we only got 2 individuals from that family. And we covered quite a diverse array of genre across the family as well. So our smash and bash approach worked pretty well. And so our first question was, does climate shape heat resistance in native bees? Yes, it does. And so we found here, unlike Drosophila, we found that maximum temperature of the environment was the most important environmental variable. And so bees that live in hot environments are more heat resistant than bees that live in cool environments. So we then wanted to ask that question of whether or not we could find any structure across bee phylogeny. And so it was a little bit surprising. So unlike what we'd found for Drosophila, we didn't really see any structure. And so we've kind of been hypothesizing, does that mean that perhaps there's greater capacity for bees to increase their heat resistance? This idea of behavioural thermoregulation was touched on earlier today, but we were also really interested in this idea. We knew that bees, they have these really diverse nesting strategies. And so we hypothesized that bees that are ground nesters, they're probably going to be less heat resistant because they have this microclimate. If it's a really extreme hot day, they can just go and hang out in their nests and be completely buffered. And then we thought our wood kind of cavity nesters would kind of sit somewhere in the middle, and then we'd have our pithy stem nesters that would be the most heat resistant because they're not getting buffered, and selection is acting more strongly on them for heat resistance. And it was one of those ones of, yep, that's exactly what we found. So our pithy stem nesters were the most heat resistant. And so it kind of poses this interesting question though. So does this mean ground nesters are the most vulnerable? Because if you think about when we were estimating climate change risk, that would mean that you have more heat resistance and maybe you're more at risk or more vulnerable to climate change. But you also have these microclimates that you can go and hide in when things are really extreme. I think the answer's kind of complicated. I think, yes, in the short term, having these microhabitats are really, really important. But I think in the long term, they're actually not getting selected upon for heat resistance. And we call this the Bogut effect. So it's almost like a selection shadow, right? So they can only hang out and hide in their ground nests for a certain period of time. At some stage, they're going to have to come out and forage. So I think it's a bit of a tricky one to say. And some of you might be thinking that what about phenotypic plasticity? You were telling me before that if you develop under warmer environments, you have higher heat resistance than if you developed under cooler environments. So perhaps maybe the reason that you're seeing this lovely relationship between maximum temperature of an environment and heat resistance is simply because bees are plastic. And so this is a really hard thing to kind of get at, with the Drosophila, that's all under common garden conditions, right? So we know that the differences that we see between those species are genetic, but because we can't rear these in common gardens, we have no idea. And it's been something that I've kind of been trying to come up with ways that we can try and tease apart these environmental and genetic effects on phenotypes. And there's no real perfect solution here. But one of the things that we did do is that we said, okay, let's estimate heat resistance. And this was just in Melbourne. Let's estimate heat resistance from when they start kind of becoming active in November. So remember, it's Melbourne, so this is just coming out of our winter. Let's estimate heat resistance across the summer and see whether or not we see evidence of phenotypic plasticity. So our hypothesis being that if they're hugely plastic, then bees in the summertime should be more heat resistant. And as you can kind of see from this, and this is just individual bees that we estimated heat tolerance on, not divided by species, but overall, we don't really see this trend that we thought we would see if phenotypic plasticity was really important. And this, we were able to divide this into some of the species that we collected across the month. So we have about 8 species here. And I probably should also say, we're still figuring out species IDs. So a lot of this is still kind of preliminary. Yeah, it's been a giant challenge. So what we see here, anyway, out of the 8 species that we have, 3 species do actually increase their heat resistance across the summer, suggesting that they might be responding via phenotypic plasticity. 3 species did nothing, and 2 species actually declined. So it kind of suggests to us that yes, there might be some species that can respond via phenotypic plasticity, but overall, we're not seeing huge amounts of plasticity. And I should also say, we can't rule out the possibility that we've got selection occurring across the different months as well. So this isn't purely phenotypic plasticity. But another thing I also wanted to highlight, if we just look at this one species that increased across the summer, we found it increased about 2 degrees. And so if you remember back to the Drosophila work, our developmental acclimation stuff could only shift heat resistance by about 1 degree if we were lucky. And then if you have a look just across these species here, and this is just 8 species, we're actually seeing quite large amounts of variation in heat resistance. So from kind of 42 to 47 degrees, the type of variation that we don't really see in Drosophila. So I think bees, in terms of their heat resistance, are definitely doing something different. Okay, but this question of pulling apart environmental and genetic effects just kept gnawing at me. And so we decided to turn to our Australian stingless bees to try and help us understand this question. And so we have, as I mentioned at the start, we have about 12 species of stingless bees. They are eusocial, so they have queen and they have their workers. And they tend to occur kind of Sydney upwards. So I'm really jealous. Everyone that works on bees in Sydney and above, they have their own little stingless beehives in their backyard. Maybe climate change will have some positives because I can get a stingless beehive. Anyway, and the great thing about stingless bees is that they are in a box, and so we can pick up those boxes and we can move them. And so there are 2 ways that you can get at genetic and environmental effects. You can do common garden experiments like what we did in the Drosophila, or you can do a reciprocal transplant experiment. And so this work was all done by a really amazing master's student. And so what she did is she picked up 15 hives from Sydney of Tetragonula carbonaria, and she took them on a summer holiday up to Tweed Heads, and then she took 15 hives of Tweed Heads and took them down to Sydney. And of course, we had controls hives that remained at each of the sites. We really wanted to do the reciprocal transplant on the full distribution of Tetragonium carbonaria, but this is COVID. So during this experiment kind of happened towards the end of 2020 to 2021. And during that time, we were having lots of issues with COVID, and sometimes the borders would close. So we just couldn't take the risk that the border between New South Wales and Queensland would close during the experiment, and then we wouldn't be able to get to our hives. And indeed, that is what happened. So it was a good thing that we didn't do that. And so the hives were out at their different holidays for a year, and we estimated heat and cold resistance. We also estimated some fitness traits, but it turns out fitness traits are really hard in stingless bees, and they kind of were basically the subtropical tweed heads had more flowing resources, so all the bees that went on a holiday up there were just much happier. And so I think if you wanted to look at kind of fitness in these eusocial bees, you'd need to kind of track across generations, I think, a lot longer. And so we estimated the heat and cold resistance 4 times across that year as well. And so our predictions were that if it was genetic, so if divergence between heat resistance between these two populations was genetic, then your Sydney population at Sydney and in subtropical tweed heads would look the same. So there'd be no differences between the populations. But if it was plasticity, then we thought that our Sydney populations would then start to look like our tweed head populations, and our tweed head populations would start to look at Sydney in their away sites. So what did we find? Not a lot. So it didn't really matter when we estimated heat resistance, whether it was just post winter or whether it was in the middle of summer. There was no divergence between the populations, and there was no phenotypic plasticity. And we actually did find some really kind of fun results for cold resistance, but this is a heat talk, so I can't show those. And so we're like, why? We started kind of ponder why this is the case. And so if any of you guys kind of know Australia, it's getting increasingly warmer across the east coast of Australia, and so this species stops around Brisbane. And so we think that maybe heat resistance is actually limiting the capacity for these species to shift further north. And we do have some other tetragonial species that we've estimated heat resistance in that have these more northern distributions, and they have higher heat resistance. We also suspect that maybe competition from those better heat adapted species might also be playing a role there as well. But we are still trying to figure out ways that we can actually test that. So just kind of contrasting that question of what do the results that we see in Drosophila, are they also the same in bees? And I think the answer is kind of yes, but you know, with a few differences as well. And so we found that CT max is linked to the environments in both bees and Drosophila. And I think that's really important. And I also want to make the point that, you know, we're very much about trying to find proxies of climate change resilience. And we really need to understand or know that whatever trait we're using to try and understand this has some kind of predictive power now. So it has to be able to explain something about where species are living currently. And I think we can say that, you know, CT max has a few issues, but at least, you know, this research shows that it does have some predictive power. We found kind of both in the Drosophila and the native bees that perhaps there isn't that much plasticity happening in heat resistance. We really saw that in the stingless bees with our experiment there. I think there's more work to be done on plasticity in the bees, right? Because it is a really hard thing to kind of untangle when you can't rear them in the lab. And we found these differences in the capacity of bees and Drosophila to increase their heat resistance. So we found this strong kind of phylogenetic signal in Drosophila, and we didn't see that in the bees. So, like, tantalizingly, maybe this suggests that native bees have greater capacity to increase their heat resistance when compared to our Drosophila species. And I just wanted to kind of end the talk kind of thinking about these warming margins and these predictions of risk as well. And so these warming margins have kind of been heavily kind of debated about how useful they actually are. And I think the absolute values of warming margins are perhaps not that meaningful, because if you're in the heat resistance world, you know that your estimate of heat resistance will vary depending on how you actually go out and estimate it. But I think where these warming margins can be useful is they can be useful in making these kind of comparisons between species. I think, you know, it's quite kind of surprising how much more resistant bees are in comparison to Drosophila. And I suspect that is something to do with their ecologies. So Drosophila, they're hanging out in the dirt and they're eating rotten fruit, and they're active in the morning and they're active in the evening. And our native bees, they're in the middle of the day foraging for resources. So I think this is really important. And I think we also we can still use these warming margins to make predictions about which types of species and environments might be at greater risk to climate change than others. And I think one of the things that kind of comes out of these warming margins is that our subtropical environments tend to have lower warming margins than our tropical and our temperate environments. And living in Melbourne, like, that's not very surprising to me. Some people often think, oh, well, surely tropical environments. But in Melbourne, where we have these kind of subtropical temperate environments, we experience those really big extreme events, right? So we have much hotter summers in comparison to kind of stable Cairns. And so I think that's probably why we're seeing these patterns that subtropical environments have the lowest kind of warming margins. And I think that is it. There's a lot of people that have been involved in this work, both the Drosophila work and the bee work, that need thanking. And yeah, a big shout out also to Carmen De Silva, who collected all the trait data in the bees.
Thank you very much. We'll take questions. I'll try and take questions from ECRs and students first. We've got a roving mic, so please wait for the mic to come. We've got, we'll take Becky right at the back. We'll give you some exercise.
Thank you. It's really interesting to kind of compare the issues that bees are facing in Australia compared to like native bees in the UK. Usually when you're trying to find native bees in the UK, you're limited by cold and the wet weather. And one of the things we kind of think about is body size and how that can impact thermal sensitivity and when the bees come out. So I was just wondering, do you see any signs of body size in Australia?
Yes, that is something that we're doing. And so my postdoc is still kind of pulling things of bees to try and do the body size data on that. So it's still a little bit unknown. Anecdotally, you know, I'd say that we find these kind of chunky, kind of big bees tend to be more kind of up north, which kind of goes against that kind of Birdman's rule idea that you think you'd see lower body size in warmer environments in comparison to cooler environments. So I'm going to have to go back to the Drosophila literature and say that we don't tend to find strong associations between body size and heat resistance in those species. But we don't know yet for the bees, but it's coming.
Thank you for your talk. I love it. I was just wondering, because you're comparing like natural populations of bees and lab reared population of flies, as Drosophila, and can it influence like its resistance and the plasticity? For example, the differences you're seeing in heat resistant plasticity?
Yeah, I think that's a really interesting question. I think there's kind of two elements. I mean, some at least the stuff that I've done on the phenotypic plasticity, they tend to be collected from kind of natural populations, and we like to try and estimate traits pretty quickly. I can't say that about the 100 Drosophila species. They were a lot of stock centre lines, but all the plasticity stuff is done on fairly freshly collected Drosophila. But I think nutrition is probably going to play a really important role, and we know that different labs that have different Drosophila food get different results on traits. Right, so I mean that is really possible, and particularly when you're comparing across different species where you have different nutritional requirements. And so I think it's definitely possible, but really hard to kind of expand out and ask that question.
Juliet, just here, and then we'll go to David about this.
Thank you, lovely talk. With the stingers bees, we know bumblebees and honeybees will thermoregulate their nest to keep a very constant temperature for the brood. And I think there's evidence of resilient stingless bees thermoregulate. Is that likely to affect their heat resistance as adults, or do you think that doesn't matter at all?
We don't. There has been a little bit of work done on sparing stingless bees, and we don't think they're regulating their nests the same way that the Brazilian bees are. So we don't think it's important, but I think probably there's more work that needs to be done in that space to really understand what it is.
But I mean, was it maybe?
Hi there, thanks for a lovely talk. I'm interested in the plasticity, because looking at the CT max, that is a ceiling trait.
Yeah.
So I guess you're not really going to be surprised that you're not going to see that much plasticity in that kind of trait.
Oh, I would argue it's not quite a ceiling trait the same way that viability or something or fitness related traits are. I think, you know, there should be capacity like for them to still be able to shift their critical thermal maxima. So I don't think that what you're saying is that, so when you have these estimates of kind of viability where you can only be one, like you can only be 100% viable, you know, often people kind of say that you have low plasticity when you're 100% viable, but you can't actually be more plastic. And that's
well, no, because what I was going to go on to say was that I could imagine that, I mean, if you're looking really at its absolute maximum, that is going to be a ceiling, but sort of in the little bits below that, and then in an organism will experience fluctuating temperatures across the day, across seasons, and they will absolutely be phenotypically plastic in terms of balancing metabolic costs and all the rest of it, not just day to day, but moment to moment. So they will be plastic, and there'll be a lot, and potentially be quite a lot of plasticity in dealing with their moment to moment thermal environments. But I think the CT max sort of, it doesn't surprise me that perhaps has less plasticity and doesn't necessarily map to their greater plasticity in terms of metabolism and of course behavior that they would deploy to actually deal with temperatures in the wild.
Yeah, I'm trying to think back because there has been some stuff done on metabolic rate in Drosophila as well, but it's making me kind of, I don't know if anyone's looked at their kind of metabolic plasticity in terms of rearing them under different temperatures and looking at how plastic metabolic rate is, but metabolic rate is also a complicated trait. Because what is a good metabolic rate? I don't know. So I don't think I know the answer. I think we might have to agree to disagree about the capacity for plasticity in nutrition.
It's actually probably a related question to that one. I often wonder if we miss a trick in some of this heat tolerance, I should say, cold tolerance.
Yeah.
In that we tend to look at a signal maximum value of some sort. What happens if we look at intermediate sublethal values? And you think, well, that's the space they're going to encounter before they ever get to the maximum. So if you are going to find either plastic or genetic influence on performance, maybe we should be looking 2 or 3 degrees below the CT max for some percentage. Yeah,
I absolutely agree with you. And that's one of the criticisms of CT max in general. And so if I get funded, which I really hope I do, our plan is to actually start looking at sublethal effects So we want to look at foraging thermal limits, and so we want to go out into the field with thermal imaging cameras and kind of measure when bees are active and look at their capacity to behaviour and physiologically thermoregulate. Because another thing that kind of really shocked me about bees is that you also have facultatively endothermic in there as well. So they can have some they can elevate their body temperature, and they also can physiological shunt their hemolymph from their thorax to the abdomen. So they can do a whole bunch of amazing things to kind of regulate their body temperature. So I think going out into the field and trying to get an idea on some of these sublethal limits, and then asking that question, how well do these critical thermal maxima actually predict these sublethal limits? And I think that will help us understand if CTmax is actually a really good trait for predicting some of this stuff. Yeah, I need funding.
Time for a couple more questions. Anyone has them? Nice one then. Is Drosophila melanogaster just super weird and it's expanded out of Africa, super cosmopolitan? Do we need to see these sort of selection experiments from other species to try and see what's really happening in a sort of more typical insect?
Yeah, I think so. Yeah, that's the thing that we really like to do, is start kind of these selection experiments on mesotropical species, but also the real extreme kind of desert adapted species as well, and see how far we can actually increase people within those species. And perhaps mountains just, good model for some things, but maybe for understanding climate change, resilience might not be. It's not the norm, right? Most species don't have this global distribution that melanogaster has. Yeah.
It was really, really good at last time as we spoke this morning. My mind is worrying, so I'm obviously thinking of practical implications of what you're doing. And I was looking at plasticity on your list, and it's good to know that it's thermal temperatures, and it's not going to be able to tolerate much more than 37. However, at that point, we probably won't be able to grow the crops that it's invading. So I'm wondering, in your future research, if you're going to start looking at phenotypical mismatches between the resources that they're relying on, because regardless of what they do, if the food source, for example, isn't there, then that's obviously a big threat. And the other thing I was thinking about was bumblebees as well. So I think you have Bombus terrestris now in Tasmania, and I don't know if it's in Australia.
No, I don't think it is. It's not on the mainland. We're very much trying to keep it off the mainland.
Okay,
yeah,
that might be another. So that's another species that I work with that we have real issues with in tunnel crops, because you get up to 40 degrees in the middle of the day. They spend a lot of time then farming the brood. So there are some real practical implications in what you're doing. I just wonder what your thoughts are on that, and whether that's where you see this benefit going.
I think in answer to your first question, we don't really understand the ecology of Drosophila. And I think that's a really a big shortcoming of working on the Drosophila system. So I don't think we really are in a position to kind of start thinking about kind of those mismatches between kind of food resources. I mean, I think they are just fairly generalist species, although I always say that one of the surprising things is that we see Drosophila melanogaster, it will butt up against the tropical rainforest, but it will never catch melanogaster in the tropics. So like, there must be some kind of resource differences going on there, but we just, we don't know. In terms of kind of translating this research to the next kind of, I guess, step, and that would be great, but I don't think I'm there yet, and I'd love, like, if people have suggestions. I think I'm very much a lab junkie, sitting in the lab and doing these things, and trying to think about how we can take these results and then put them into kind of more policy, or I think that would be great. I'm not sure how to do it.
Really good opportunities for collaborative research.
Absolutely, yeah, absolutely.
Brilliant. Okay, well, I think we're going to do a little closing thing, but first we're going to thank Vanessa and all the speakers today. So I'm going to quickly wrap a few things up on behalf of the local organising committee. So myself, Ben Ogden, Ben Raymond, and Jason Chapman. So firstly, I've been asked by Luke that you're going to receive a form from the Royal Ento Stock in the next few days to give constructive feedback about anything you think can do differently or better at future Ento conferences. So when you get that, please do take a few minutes to fill that in. We need to do a few thank yous. Firstly, to the amazing RES team who put this conference together. In particular, Luke and Amy, who are sat down at the front. So let's give them a big round of applause. Secondly, thank you to everyone for coming all the way down to Cornwall. It's been lovely to see you. We've had a fantastic set of presentations. So let's do a round of applause for everyone for coming to Cornwall and for doing great talks. So that's all from me. I'm going to pass on to Helen Boy now as Vice President of the meeting.
Thank you very much. First and foremost, Jane apologises very much that you have me saying thank you to everyone and that she's not here. She would very much like to be stood here. So on behalf of Jane Hill and all the Royal Entomological Society trustees, thank you to the amazing, amazing conveners, Ben and Jason. It's been an amazing programme. It's been really fantastic. And to be down in Cornwall, which is where my family are from actually, has been amazing for me, but I think it's been incredible for everyone. I think you have really showcased the University of Exeter down here on the Penryn campus. So thank you all so much. And thank you so much to all the student helpers. You have been amazing, and seeing you out at the moth trap this morning and then still running around and being really energetic throughout the sessions has just been fantastic. But thank you for the moth trapping experiences as well. It was really fun being there with you all this morning. I want to join you, Ben, thanking the Royal Entomological Society team, and again, Luke and Amy, of course, you've been amazing, but all of the staff and everyone who's here, but also all of the staff who have been online as well. But yeah, you know, Amy, you've only been here with the Royal Entomological Society for a very short while, and yet you have been absolutely incredible and looked after us all so well and ensured that we've just had this really, really successful meeting. So thank you very much, Amy. It's been amazing. Of course, a big thank you to everyone here. Again, yes, to all of the speakers. It's been just all the sessions I have sat through has just been absolutely fantastic, absolutely riveting, and a really, really wonderful program. All of the posters, all of the amazing discussions that we've been able to have over the coffee breaks and dinner and other things. It's really been a really fantastic meeting, and I hope that you'll consider going to Ento24 and we'll all meet again there. And I've been told you're going to get a save the date very soon and early bird registration details. So then I have the pitch for the Royal Entomological Society. I understand that many of you are members and fellows, which is really fantastic. But I also understand that some of you aren't members and fellows, and it would be really wonderful if you would like to join the community. And I guess that gives me an opportunity to personally say thank you to everyone. So I'm now no longer a trustee. I have completed my 4 years as a trustee, and it was a really incredible experience. And I want to thank all of you and the wider Royal Entomological Society community for making it that amazing experience. And if you're not a member or a fellow, I really, really urge you to join. You know, people will say, so what do you get from the society? And there's many, many things we get from the society, but I think perhaps the most wonderful part is that feeling of being a community and the way in which we can reach out to one another. And of course, we could do that if we weren't members and fellows of the Royal Entomological Society, but there's something very, very special that we come together through the Royal Entomological Society as an amazing entomological community. And you can find out more on the Royal Entomological Society website as how to join if you're not already a member. And it is a really exciting time to be joining. I mean, obviously, you've seen over the last few years the changing in the branding, and I'm sure you've read the strategy from the beginning to end, and you'll see of the aspirations and ambitions the society have the restructuring of some of the committees and the opening of those positions to everyone here, please get involved with society. You'll find it phenomenally rewarding, and the society will benefit enormously from you as well. So if you want to chat to anyone about what that might look like for you to be involved more within the society, I'm sure that those who are here and trustees or committee members or even outgoing trustees would be very, very happy to speak to you about that. But it is a very exciting time for the society, and I look forward to your involvement in it. There's so many opportunities through the journals, through committees, through meetings. So please get involved. So just to finally say, for me, it's been wonderful to be down here. It's with sadness that I'm stepping away now from being a trustee, but I'm really excited to be watching what happens next with the society, and I will enjoy very, very much being a fellow and meeting with you all. But my special thanks finally again go to the organisers of this meeting. I have loved being down here, and it's been wonderful to be with you all, and it's been made possible because you put so much work in. So thank you, have a wonderful journey home, and see you all very soon.